In this particular figure, we're looking at consequences of non-disjunction, which is failure of chromosomes, either homologous chromosomes or sister chromatids, to separate during meiotic divisions. Now remember, meiosis is the type of cell division that is associated with the production of gametes, meaning sperm or egg, and gametes have the haploid number of chromosomes. So in other words, there's a reduction of chromosome number as well as a reduction of chromosome material in order to produce the gametes. So in this particular figure, we're looking at a pair of homologous chromosomes. The color distinction, dark versus light, meaning that this is one member from mom, one member from dad. Each of these chromosomes has duplicated in preparation for cell division, in this case meiosis. So I have two sister chromatids for this member of the homologous pair, two sister chromatids for this member of the homologous pair. Now, there are two division events in meiosis. The first meiotic division, which is called reductional division, and the second meiotic division, which is called equational division. Normally, reductional division involves the separation of the homologous pair of chromosomes. So you reduce the chromosome number. This means we go from 2n, right? In this case, 2n is equal to 2. I go from 2n to 1n. In other words, the first meiotic division reduces the chromosome number by 1/2. So I go from diploid 2n to haploid 1n. So the question now becomes, well, why do we need to go on to a second meiotic division? Because I already have haploid cells. The answer is the problem is the amount of genetic material. In other words, each chromosome is still replicated. You have two sister chromatids, and what we need is one member of the chromatid pair. Otherwise, we've got too much DNA. So let's look at the normal scenario first. In meiosis 1, reductional division, we normally have separation of homologous chromosomes. Meiosis 2 is called equational division because you don't reduce the chromosome number. Here I have one chromosome. It is duplicated, but it's only counted as one chromosome. So when sister chromatids separate in the second meiotic division, each of my resulting daughter cells has one chromosome in it. So this is why it's called equational division. I have one chromosome at the start, I have one chromosome but unduplicated at the end. Again, one chromosome duplicated at the start, one chromosome unduplicated at the end. So I haven't reduced the chromosome number, I've just split the sister chromatids. Okay, so if I have fertilization take place, then if this is an egg, for example, then a sperm that has one chromosome will cause fertilization to occur, and in both of these cases we would end up with the normal diploid number. Now remember, these chromosomes are not duplicated yet, okay, in preparation for the next division event that would occur. So this is the normal scenario. Meiosis 1, we reduce chromosome number. Meiosis 2, we separate sister chromatids. Fertilization, we restore the diploid number. Now let's look what happens if we have non-disjunction in the first meiotic division. Excuse me. In this case, we have failure of homologous chromosomes to separate during meiosis 1. So this daughter cell has an excess chromosome in it. This daughter cell has no chromosome in it for this particular pair. Now, if we only had one pair of chromosomes, then this would never develop. Okay, but what the assumption is that we're talking about humans. There are 22 other pairs of chromosomes, so the loss of a particular chromosome, in other words the monosomic condition, is not necessarily fatal in terms of embryonic development. So in this case, I'm -1 with regards to chromosome number. If fertilization does take place, then I will be monosomic for this particular chromosome. Okay, there would still be the other normal chromosomes there, but I would be monosomic for this particular chromosome. Now, in the other daughter cell that got the extra member of the homologous pair, in reductional division, the homologous chromosomes are normally supposed to separate. Here they didn't. So now I go into the second meiotic division, which is the separation of the sister chromatids. So I get one daughter cell that has a light and a dark, another daughter cell that has a light and a dark. Now what has happened is I have an extra copy of each one of these chromosomes. So at fertilization I now become trisomic for this particular chromosome number. For example, trisomy 21. The condition of monosomic, for example, would be Turner syndrome, where there's only one X chromosome present in the particular fertilized egg. So in the situation of non-disjunction at meiosis 1, you get either trisomic, after fertilization, you get either trisomic or monozoic fertilized eggs. Now what happens if we have non-disjunction in meiosis 2? Well, we've already seen that if I have that scenario happen, one half of the fertilized egg should be normal diploid, and we already went through that earlier. What happens if I have non-disjunction of the sister chromatids? Well, both sister chromatids go one direction and no sister chromatids go the other one. So what I'm going to end up with then is a monosomic situation, where over here everything is either trisomic or monosomic. Over here half of the gametes are either trisomic or monosomic. So when they're saying, by the way, I should have mentioned this earlier, this normal gamete is what's doing the fertilization. So we're saying in each case this would be fertilizing this would be fertilizing this would be fertilizing this, and so on. So I should have mentioned that a little bit earlier. So the consequences of non-disjunction in meiosis 1 are all fertilized eggs are abnormal. If you have non-disjunction in meiosis 2, only half of the resulting fertilized eggs are abnormal.